Welcome to Canine Cancer Alliance webinar. My name is Mary Maeda. I'm the director of Canine Cancer Alliance, which is a nonprofit in Seattle, but our volunteers are everywhere around in different states. And our main thing that we do is provide funding and assistance to researchers, research to support clinical trials to find cures for cancer in dogs. So the nonprofit was started out of our frustration with today's therapies, and hopefully we will find something that is safe and effective to help our dogs. So today's webinar, I'm going to talk about immune checkpoint inhibitors. What are checkpoints? What are these inhibitors? I'm going to talk about human medicine, but just give you a background. But there are some very exciting developments in veterinary medicine. So, you know what? Before I start, I should mention that our nonprofit is made possible because of donations, and we're looking for donors who are monthly donors, if it's possible, but 100% of donations that we receive goes to support research, such as the EGFR HER2 vaccine, clinical trials, and other projects. And also, in a little while, we will be starting accepting submissions for our calendar for next year, 2025 calendar. And this love calendar started last year, and we would like to get every day of the year filled with pictures of your lovely dogs. So we will be announcing that very soon. I just want to start out by sharing the fact that about 9 years ago, President Jimmy Carter went into complete remission when he had stage 4 melanoma. So stage 4, that means that there was metastasis. Cancer had already spread from his skin to brains and other locations, but he was totally cured. And that was thanks to immunotherapy treatment that he got, a type of checkpoint inhibitor. So, what is immunotherapy? Instead of trying to kill cancer cells directly with things like chemotherapy or radiation that indiscriminately kills cells, healthy and cancerous, immunotherapy is helping natural immune system find and kill cancer cells. So, for example, in this picture, you see a cancer cell on the top with a T cell on the bottom, and T cell is there are many immune cells, but T cell is one of the more important ones, and it can actually target, find cancer cells, and destroy it. Now, I want to do a little bit of historical excursion just to let you know that there were some inklings that immune system was playing a role in our fight against cancer. There was Rudolf Virchow, who realized that there are pus cells inside tumor and kind of linked the inflammation with cause of cancer. Back then, they didn't really have a good model for immune system, but there were some understanding of what was happening. Paul Ehrlich also, he was noticing these, what's now known as immune cells inside tumors, and he was wondering, are they bystanders or actually actively involved in attacking cancer? And so these clinicians and researchers were starting to be a little bit involved. And then in around end of 19th century, William Coley, who was a surgeon in New York City, he invented a bacterial extract actually to successfully treat cancer patients. And what happened in his case was that he noticed that there were sarcoma patients who had very advanced sarcoma, tumors were growing, but they became cancer-free after experiencing a bad skin infection. And at the time, he didn't know the cause, but he knew that if you get fever, sometimes cancer goes away. So he actually created a bacterial extract concoction, and they called it Coley's extract or Coley's toxin. And he began treating cancer patients that way, many sarcoma patients. And by the way, today it's not possible to cure sarcoma generally. But back then, over 100 years ago, he was successfully treating and curing about half of his patients, according to the records. And that was using this immunotherapy, using bacterial extract. Now, unfortunately, Coley's work sort of got shoved aside because radiation therapy became popular and chemotherapy was introduced. So about 50, 60, 70 years after Coley's invention, Coley's toxin was not used, and clinicians mostly depended on chemo and radiation therapy and, of course, surgery. And this Robert Schreiber, who's an immunologist, he stated that in that time, immune system, most scientists, researchers said that immune system plays no role in treating cancer, and any scientist who thought otherwise was wasting valuable time and energy. So unfortunately, even though they kind of were building up an understanding how the immune system worked, they didn't think immune system was involved in killing cancers, because if it was involved, why isn't the immune system actually taking care of cancer? So it turns out that cancer can put brakes on the immune response. And even though you may be able to activate the immune system, cancer responds by putting brakes on it. And that's what I'm going to go talk about today with immune checkpoint inhibitors, because immune checkpoint inhibitors are the kind of drug that actually cancels out, inhibits ability of cancer to put brakes on. So in this picture, I show an example of checkpoints. So checkpoints are molecules on immune cells that can either turn off or on the immune response. And you see an alphabet soup here: PD-1, CTLA-4, LAG-3. There's many checkpoints on the surface of immune cells, such as T cells. And here in this example, you see T cell interacting with a cancer cell. And on the surface, T cell has a PD-1 protein expressed. And when the ligand, cancer cell, expresses PD-L1, and when they come together, it inactivates the T cell. So PD-1, PD-L1 interacts, an immune cell, this case T cell, is inactivated. So T cells have an off switch, and cancer can use that off switch for self-preservation. And why do they have an off switch? Well, that's because when immune system gets activated in response to infections, you need a way to make sure it doesn't just go and turn into a runaway immune response. So you have to be able to turn on and off the immune system. So these checkpoints naturally exist on the surface of the immune cells, but unfortunately, cancer has figured out how to use it for its own survival. So the good news is that the hard work of researchers in many different countries, but in particular, a Japanese researcher at University of Kyoto named Tasuku Honjo and Jim Allison discovered two very, very important checkpoints. One is called CTLA-4, and the other one is called PD-1. And they're involved in creating a therapy that basically blocked cancer's ability to use these checkpoints. And they were awarded Nobel Prize. So just to illustrate what's happening with example of a PD-1 checkpoint, on the very left here, you see a T cell, and it's activated, but then the tumor cell comes with its PD-L1 ligand, the protein on the surface, and when they come together, T cell gets suppressed. And so cancer is no longer being attacked by T cells. Now, but if you have antibodies, either anti-PD-L1 or anti-PD-1 that blocks PD-1 molecule, then you could actually stop this action, stop the immune suppression by cancer. So that's what checkpoint inhibitors are. It's usually these monoclonal antibodies that blocks PD-1 or PD-L1 or other checkpoints. So there has been thousands of clinical trials involving human patients, and CTLA-4 blockade, an example of a drug is ipilimumab. They saw that with the help of this drug, CTLA-4 blockade drug, very advanced melanoma patients, and they can see a response where you can see a tail develop. So this is a Kaplan-Meier survival curve, and what it shows is the number of patients, a percentage of patients still alive, surviving over time. So unfortunately, the number does go down, 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 down. But what they saw was as the number went down, it sort of flattened out and plateaued out into this tail. And that's the magic of immunotherapy. These patients, 20% or so of the patients, were living long time, effectively cured, like President Carter, even though they had metastatic or very advanced cancer. And so this drug, CTLA-4 blockade drug, was approved by FDA in 2011, and other checkpoint inhibitor drugs started to become approved by FDA for, of course, human use. Now, the limitation is that it's only successful. The treatment is only successful in minority of patients. Initially, it was like 10, 15, 20%. And it was successful only in subset of cancers, especially melanoma and cancer types that seem to be associated with more mutations. They call it TMB, tumor mutational burden. And also, there were side effects. Even though you're not directly killing healthy cells, when you activate the immune system, there's always a risk that immune system will attack some from healthy cells. So there's autoimmune response and inflammation. So those are the limitations. But researchers realized that if you start combining some of these immunotherapies or checkpoint inhibitors, like combining the CTLA4 and PD-1 blockade drugs, then you can get more patients to respond. So for example, here, the bottom shows CTLA4 blockade drug, and about 26% of human patients were surviving until 5-year point. Now, the other drug, PD-1 checkpoint inhibitor, nivolumab, had patients surviving, about 44% of patients surviving. But then when they combined it together, over 50% of the patients were alive at 5-year point. So the key is combining different therapies, especially immunotherapies, could lead to much better efficacy. So now in human medicine, there are many checkpoint inhibitors that have been approved by FDA, and you have to get approval by cancer type. So CTLA4 and PD-1 checkpoint inhibitors, et cetera, have been approved for melanoma, and now lung cancer, kidney, colorectal, lymphoma. So all these checkpoint inhibitors are getting approved. Now, actually, if you look at it carefully, this is not many different kinds of checkpoint inhibitors. It's only like 3 or 4 different kinds of checkpoint inhibitors, but they are getting approved for many different types of cancer. So how about immunotherapy and immune checkpoint inhibitors for dogs? Now, if your dog has cancer now, or in the past, if your dog did, then you might be aware of some of these immunotherapy treatments that are available for dogs. Immunocytine is one, and this is probably the oldest one that's been around. It's a mycobacterial cell wall fraction. It takes a cell wall of bacteria, which is known to stimulate the immune system, and they made drugs out of it. So it's not live bacteria, it is dead pieces of bacteria. But it was approved by the USDA for treating mammary tumor, but some vets are now starting to use it for other types of cancer, including, well, quite a few different other kinds of cancer. Oncept vaccine was approved probably about 15 years ago, and this is only for oral melanoma. It's a DNA vaccine. Torgen and Ardent vaccines are made from the cancer tumors that you send to them, and they'll actually create a vaccine for your dog. So it has these cancer vaccines to stimulate the immune system. EGFR HER2 Theragen vaccine or EL vaccine is a peptide vaccine that's undergoing clinical trial right now. It's available at 10 clinics around the country, and it's showing promise, especially for slowing or preventing metastasis and osteosarcoma, and perhaps even for bladder cancer and hemangiosarcoma. Elias immunotherapy is another immunotherapy where they activate dogs' immune system by giving them autologous vaccine, taking the T cells out, reactivating it, reinfusing the T cell back in, and then giving them IL-2 cytokine. And they've been running large clinical trials for osteosarcoma patients, and I believe that they're also going to be looking at other types of cancer. So these vaccines have been available or have been in trial or have been approved, but just recently, immune checkpoint inhibitors have started to become available for dogs. And I'm going to take a little bit of time and talk about GIlvetmab, but there are also other checkpoint inhibitor drugs as well, which I'll touch on. So I mentioned PD-1 and PD-L1. Cancer expresses PD-L1, so it can attach to PD-1 on T cells and inactivate it. So which types of cancer actually expresses PD-L1? There have been studies that looked at tumor samples to see how much of expression there are in different kinds of tumors. And so here, Mayakawa, a Japanese study and a different study, they looked at many kinds of tumor cell samples, and osteosarcoma expresses PD-L1, nasal adenocarcinoma, melanoma, mammary gland carcinoma, so there are lymphoma as well, that express PD-L1. So they might respond to a checkpoint inhibitor that blocks PD-1 or PD-L1. Now, the last 3-4 years, there have been quite a few publications, a lot of data published by Japanese research groups, and they looked at anti-PD-1 or anti-PD-L1 checkpoint inhibitors, and they looked at melanoma and advanced cancer types. And there was a lot of data published, and it was very exciting. And here, for example, here's work that was published last year that showed that if you gave dogs radiation therapy and immune checkpoint inhibitor this is melanoma patients, I think, that if you don't give them checkpoint inhibitor, you see this black curve. So that's the survival for these patients. But if you give them checkpoint inhibitor, especially when radiation therapy is given before, then you can get this red curve. So the survival time can be extended dramatically for a subset of patients. So these results are really exciting, but these are all studies in Japan. And it would be great if they could make the drug available in the US, but we didn't really hear that anything was happening, commercialize it. And to commercialize a drug, to make the drug available widely, it has to be licensed by either FDA or USDA. For veterinary drugs, FDA approves small molecule drugs, but USDA approves most of cancer immunotherapy drugs, including vaccines and monoclonal antibodies. And so are there checkpoint inhibitors that are going to be approved by USDA? And just to let you know, usually researchers and clinicians have to take a new therapy, first run small pilot clinical study, and then expand the study, and then hopefully they'll get conditional approval from USDA that says, okay, now you can start selling these drugs, make it available to a lot of dogs, but it's not fully approved yet. And you have to run something called pivotal study with many dogs and get full approval. And just to give you an example, Elias immunotherapy for osteosarcoma now has full approval from USDA. It took many years. The Yale vaccine, EGFR HER2 vaccine, is still trying to get conditional approval. They have a lot of data, but there's a lot of administrative, bureaucratic impediments for getting USDA approval. And GIlvetmab is a drug that was recently conditionally approved by the USDA. So it's a checkpoint inhibitor. It's a PD-1 checkpoint inhibitor, and it got conditional approval for treating melanoma and mast cell. And this is extremely exciting because this is a first canine version of checkpoint inhibitors. So let's see, I think I've said most of this, but it's a canonized anti-PD-1 monoclonal antibody. And it's a little bit different from the human version because these proteins are slightly different in dogs and mice and humans. So they had to create one that was more suited for canines.
GIlvetmab has been evaluated with 26 dogs with mast cell tumors and 25 dogs with melanoma. Stage 1, 2, 3 mast cell tumor patients, and stage 2 and 3 melanoma patients. The response rate for mast cell tumor was 73%. For melanoma patients, it was 60% according to Merck. So what do these rates mean? We couldn't find any published articles, but we did find a poster presentation by Merck research team at a veterinary conference. And it breaks down the response rate by complete response, CR, partial response, PR, stable disease, SD, and progressive disease, PD. So for mast cell tumor, 73% is the sum of dogs who experienced complete response, partial response, and stable disease. If you exclude dogs with stable disease, the objective response rate was 46%. For melanoma, 60% response rate includes dogs with stable disease too. And so if you subtract the stable disease dogs, then the objective response rate was 20%. So just like with human studies, only subset of dogs respond to these checkpoint inhibitors. No Kaplan-Meier survival curve has been published, as far as we know.
It also goes through and published sort of the different adverse reaction side effects. And so lethargy, fatigue, reduced appetite were some of the top ones, and vomiting, weight loss, anemia, nausea, and anemia. So these are a list of adverse reactions. And even though I said that immunotherapy tends to be safer than chemotherapy, which indiscriminately kills all the cells, based on human immune checkpoint inhibitor studies, it is possible for patients, many patients. So those are really serious adverse events. And with these very small studies, you really can't tell what kind of side effects we have to be concerned with. Now, so Merck released GIlvetmab last October, and they said that they would donate this drug to research teams who wanted to design a study. They had to get other funding. So Merck was not going to provide funding, but they would donate the drug GIlvetmab for the treatment. And so over the past almost 12 months now, new studies have launched that is looking at the combination of GIlvetmab with other therapies, and not just for melanoma and mast cell tumors, but for other types of cancer. So, for example, at Tufts, they're looking at GIlvetmab and another drug for T-cell lymphoma. At Missouri State University, they're looking at combining Elias immunotherapy for osteosarcoma with GIlvetmab to see if they can increase the response of those patients. There's a number of studies that's looking at combination of GIlvetmab with radiation therapy. And there's also one for bladder cancer. So there's a number of studies right now, clinical trials, that are ongoing involving GIlvetmab. But I should let you know that GIlvetmab, because it has conditional approval, it can be ordered by oncologists for anyone that they think might help. And for a while, it was out of stock, and so oncologists could not order it. But I heard just this week that they are starting to take orders. Now, I want to just talk about the last one, GIlvetmab and the EGFR HER2 vaccine study for hemangiosarcoma. And that's taking place here in Seattle. And it's a collaboration between Professor Mark Mamula, who invented the vaccine, and Dr. Chelsea Tripp, and also Professor Natalia Shazenko, who is at Oregon State University, who is an expert with gut microbiome. And so here the idea is for hemangiosarcoma patients, would the combination of chemo and immune checkpoint inhibitor, GIlvetmab, and EGFR HER2 vaccine help extend the survival times? And also, let's do the gut microbiome analysis to see if you can correlate and see who responds. Because there are human trials that have shown that gut microbiome is intimately connected to immune response, and that the patients with diverse gut microbiome tends to respond to immunotherapy much better than other patients. So with this trial, it is open now. It is enrolling patients with hemangiosarcoma, not for any other types of cancer, just hemangiosarcoma. And it is supported by Canine Cancer Alliance, and the drug was donated by Merck. Now, other immune checkpoint inhibitor trials are a CTLA-4 trial. CTLA-4 is the other checkpoint that's very famous and deeply studied and approved for human use. But now there's a company called Vetagenics, and also CSU seems to have a formulation of CTLA-4 antibodies, and they're running clinical trials. And there's also CD200 checkpoint inhibitor that showed a lot of promise when it was combined with autologous vaccine made from the dog's own cancer. So kind of really exciting that these checkpoint inhibitors are becoming available. Some of them are only in clinical trial. Others, like GIlvetmab, you can order. But the challenge is, what we're not happy with and concerned with, is that you need combination therapies to improve efficacy. Just using immune checkpoint inhibitor does not lead to cure for most patients, human or dogs. And you have to combine them. But when you start combining, adverse events start to increase. There's going to be more side effects. And also, the treatments become much more expensive. So, for example, the studies I just showed you with radiation therapy plus GIlvetmab, radiation therapy might cost $10,000. GIlvetmab, another $10,000. It's going to be out of reach for a lot of people, which we're very concerned about. So luckily, there are things that we can do about that. There are other ways to improve immunotherapy. And I'm going to mention a few here, and probably we'll have another webinar in the future to talk a lot more about ways to improve efficacy of immunotherapy. I had mentioned that there's an intimate connection between gut microbiome and immune system. And there has been many studies that says that depending on the gut microbiome of the patient, they either respond or they don't respond to immunotherapy. And so it is important to have, for people and dogs, really healthy gut. And just taking probiotics doesn't seem to help. So perhaps adding fiber. Fecal transplant actually has shown to help subset of patients who didn't originally respond to immune checkpoint inhibitor actually turn into responders. So fecal microbiota transplantation for human, that's like a big deal. You're going to kill all the gut microbiome and then reintroduce it rectally. But for dogs, there are oral tablets that you can buy from companies like Legacy Biome and Animal Biome. So could just giving these tablets to dogs help respond to immunotherapy better? We don't know, but it's probably a study that we will be supporting in the future. There are other studies, many studies that looked at antibiotics and immune checkpoint inhibitor response. So this is just one example that was recently published by researchers in Japan. And there they looked at PD-1 checkpoint inhibitor response in patients with head and neck cancer and looked at people who were under antibiotics treatment versus people who are not under antibiotic treatment. So I think they looked at like 1 month before they started checkpoint inhibitor and patients who didn't receive any antibiotics. So the Kaplan-Meier survival curve on the bottom shows, rapid, fewer and fewer patients are alive, and that's for patients who did receive antibiotic treatment, whereas other patients who may have just had antibiotic treatment like 3 weeks before it started, immune checkpoint inhibitor treatment started, those patients tended to live longer and respond better to immune checkpoint inhibitors. And this was done as a retrospective study. So it's just going through the medical records. And so it would be really great if we could do something like this for canine patients to see if they can correlate and see if dogs who didn't get antibiotic treatments are actually responding much better to immunotherapy, immune checkpoint inhibitor, or CEPT or other drugs that are available now. And this is my last slide, actually. And this slide shows a paper that just came out this month, and it's extremely exciting. And it's going to be presented next month at the Veterinary Cancer Society meeting. And this was led by a researcher from Dartmouth College Medical School. And what he did was he treated dogs with mammary tumor with metastatic mammary tumor. So the cancer had already spread to lungs. And what he did was he injected immune stimulating agent. In this case, he used a nanoparticle plant virus-based immune stimulating agent and immune checkpoint inhibitor into that tumor. And he repeated that every 4 weeks. There was a protocol. And then he could see that you could get abscopal effects. So the other tumors started to shrink, not just the one that he injected, but the other tumors also started to shrink, as well as the lung tumors also shrank. So 315 days later, this dog with metastatic mammary tumor is still alive. And many of the lung metastases, lung lesions have shrunk. One actually got bigger, but this is extremely exciting because there's a possibility to treat metastatic tumors with intratumoral injection. So GIlvetmab is usually used right now as an IV infusion, and you need a lot of the drugs to actually give as an IV infusion, like 100 milligrams or depending on the size of the drug. But if you do an injection into the tumor, you only need a fraction, maybe 1 milligram or 2 milligrams. And so that means that it's going to be a lot cheaper. You're using a lot less drug. And the other interesting thing is that the safety profile, because you're not bathing the body with the IV infusion, you're just locally injecting immune checkpoint inhibitor, you can still see a systemic effect, but the side effects tend to be milder. So this is not the only study. There are many other studies that's now starting to look at intratumoral immunotherapy, especially combining multiple immunotherapy and injecting them directly into tumors. So that's a study that we were extremely excited about, and we've started supporting project in that area. So hopefully we'll be able to share more about that at a future talk. So that's the end. Thank you, everybody.
Please like and subscribe if you found this video helpful.